To begin this unit about reproduction, we first need to look inside the nucleus of cells because that's where DNA is located, and DNA is the code for everything that's alive, and therefore that needs to be passed on in order for something to reproduce. Just a reminder, at the beginning of the year we talked about the 4 molecules of life. We have carbs, lipids, proteins, and we're looking at this 4th category, nucleic acids, today. DNA is a nucleic acid, as is RNA, but we'll talk about RNA in the next unit. It stands for deoxyribonucleic acid. You find it in the nucleus of all cells, and it's a set of instructions that essentially codes for proteins, and then those proteins can go on and do other jobs and essentially build up an organism. Do you think that you can see DNA without the use of a microscope? DNA is pretty small, but as you'll see in a lab coming up, we can actually clump it all together, and yes, you're going to be able to see DNA without having to look at it under the microscope. 4 things that you need to know about DNA. First, it's made out of 2 pieces. This is called being double-stranded. In the picture, you can see that there's this green ribbon backbone, that there's 2 pieces that wind up. The second thing you need to know about DNA is that the strands are complementary. This means that the pieces pair together in a very specific pattern. It's not just all random. In the picture, this is represented by colors. Orange and yellow are always going to pair, and red and blue are always going to pair. DNA is a helix. It's this twisted shape where the 2 strands wrap around each other. And the 4th key idea about DNA is that it's antiparallel. The 2 strands actually run in opposite directions. In my next picture here, you can see that one end is called the 5' end, the other end is called the 3'. This left strand runs 5 to 3, the opposite side runs 3 to 5. They run in opposite directions. DNA, the polymer, remember we talked about this at the beginning of the year, is like the necklace of the molecule. It's called a nucleic acid. So DNA is an example of a nucleic acid. When you hook up lots of nucleotides together, you make a nucleic acid. So the repeating beads, the things that make up DNA, are called nucleotides. I'm going to go back a picture. Each one of these, it has this phosphate group and then this ring. This is one nucleotide. Here's another nucleotide. We string up lots of these nucleotides together and you get a nucleic acid. This is a molecular picture, what you just saw when I went back a slide, of a nucleotide. Because you don't really know organic chemistry, I want you to know this. A nucleotide is a P, S, and B hooked together. I'm going to go back again. We have a phosphate group here, a sugar, and a nitrogenous base, P, S, and B. Write this down. P stands for phosphate, S stands for sugar, and in the molecule of DNA, the sugar that is part of it is called deoxyribose, hence its name, deoxyribonucleic acid. And the NB stands for nitrogenous base. We can stack these nucleotides up on each other, and you get that long chain called a nucleic acid. Here in this picture you can see phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. This is one nucleotide. Here's another one. Here's another one. They all line up, and you get a nucleic acid. Going into a bit more detail here, a phosphate is made out of a phosphorus and 4 oxygens attached. You can see that to the right. The sugar of DNA, I mentioned on the last slide, is called deoxyribose, and that's why it's called deoxyribonucleic acid. So you tack those 2 plus a nitrogenous base. We have 4 options. We have something called A, T, G, and C. A stands for adenine, and in the picture I give it a specific shape because I'm showing how these are complementary. A only pairs with T. You can see that their shapes fit. They complement each other. T stands for thymine. Cytosine is C, and guanine is G. This is not guano. Guano is a word that means bat poop. But this is adenine, thymine, cytosine, and guanine, and they pair up in DNA. I'm going to go back here. A couple more. There we go. Here's adenine, thymine, cytosine, and guanine, that they pair up and make up DNA. Who pairs with who? A pairs with T, G pairs with C. This means that they are complementary to each other. And what holds them in place is a hydrogen bond. By itself, a hydrogen bond is very weak and can break easily, but if you put a lot of them together, it is much stronger and holds the molecule intact. Chargaff's rule is something that you need to know. His rule states that in a sample of DNA, The amount of T will always equal the amount of A. The amount of C will always equal the amount of G. So, if there's 3,502,523 adenines in a section of DNA, how much thymine is there? Well, the amount of A always equals the amount of T, so 3,502,523 would be your answer. Here's another question. If 20% of a section of DNA is C, cytosine, how much adenine is there? Your gut instinct might be that this is not possible, but actually it is. If 20, and please pardon my poor writing skills with a mouse, if 20% is C, C pairs with who? Well, C pairs with G. So that means that there's also 20% of G. 20 plus 20 gives us a total of 40%. So in this sample of DNA, we have that 40% is made out of C and G. Now, we can only ever add up to 100% when we're looking at something here. So 100 minus 40 gives us what percent? That gives us 60. So 60% is left to be divided between A and T, because those are the other two bases that are equal to each other in DNA. So what you do is you're going to take 60 and divide that in half. And divide it in half, that means that 30%, wow, that's an awful 3, 30% is made out of A, and 30% is made out of T. So the answer to this problem, how much adenine? It's 30% adenine. If you don't understand that, please come and see me so I can walk you through how to do this, and we can maybe do another sample problem together. Lastly, what you should do, I have a sample of DNA here. DNA is double-stranded and the other strand is complementary and antiparallel. So please fill in, what would the bases on the other strand look like? To get you started, first we have to figure out, remember it's antiparallel, so if this is the 5' end, its complementary strand is going to be a 3'. Over here is going to be a 5'. A pairs with who? A pairs with T. T pairs with A. T pairs with A. C with G. Please finish that out for the rest of the strand, and then when you think you have the answer, come on back and play my video again and see if you got it correct. Here's the correct answer. In your answer you should have TAA, GGG, CC, TT, CTA, TCC, G. Please double check your note packet for what's coming up next, what you need to get done, and see me if you have any questions.